A recent study performed by Pablo Martinez-Lozano-Sinus, Malcolm Collar, and Renato Zanobi has revealed that the analysis of human breath may support the existence of individual metabolic phenotypes. Metabolic phenotypes show a huge range of variation, the result of many contributing external factors. This study aimed to investigate whether there are personalised phenotypes that exist despite these changing external factors, a study similar to previous investigations into urine metabolite profiling. Personalised healthcare is an important issue in today's society, and technological improvements have made significant advances towards creating medical assistance personalised for the characteristics of individuals. Genome sequencing is a significant part of this, but including metabolome into the considerations would increase the level of effective personalised healthcare greatly, as it does account for factors which affect gut microbiomes, like diet and lifestyle. However, metabolome's very nature of varying in response to external factors means that variations in an individual are present and need to be considered. This study aimed to analyse the biochemical information available in breath and investigate whether metabolic phenotypes can be stable for an individual over time. For this investigation, each of the 11 subjects exhaled into a heated Teflon tube which fed into a mass spectrometer. Each subject repeated this exhalation 3 times. In each case, the first part of the breath, which mainly consisted of air sitting in the upper respiratory tract, was not analysed. This graph shows the ion intensity as a function of time for each of the subjects' breaths in one of the data collections. MZ59, 151, and 207 represent the ions being tested, which were acetone, ethanol, and acetaldehyde respectively. You can clearly see the 3 peaks from each subject's triplicate trial. It is obvious that the amount of each ion differs with the individual, which shows that each subject has a personalised breath composition. This graph shows that there is visible grouping between the compositions of the 3 ions for each of the subjects, shown in different colours. The recognition score overall was 76%, and the inset shows how the results generated by the model match the test results between 60 and 100% of the time. This graph shows that despite the intra-subject fluctuations caused by external factors, breaths belonging to one individual were able to be identified. By collecting samples of exhaled breath and analysing their composition with mass spectrometry, the authors of this journal were able to conclude that some breath compounds can show significant variance between subjects and within a subject. But despite this variation, highly personalised compositions of breath exist and can be identified. This study shows that highly individualised healthcare is possible and that breath analysis can contribute to this future medical healthcare.